published over 140 research articles, with several studies appearing in high-impact journals such as Science, Cell, and Nature Medicine. Today's topic is about Okazaki Fragment. Please join me in warmly welcoming Dr. Binghui Shen.
Thank you so much, Shu, for the invitation. Some of you asked me if I'm an old friend of Shu. That's the reason why he invited me. Actually, I have to tell you, this is the first time we meet. Actually, online, so that's the end of the 2023. I think he's applying for graduate school, and City Hope was one of the places he was considering. But he found that my work is very relevant to what he was doing in China. And so we started to talk about this, and I feel like our mind, the scientific mind, is very close. That he was looking for those structural changes, including transpositions, impact on the function and evolution of the species. And we were more focused on the origination, how this structural variants created. And I deeply believe it's because of DNA replication errors is the most abundant resource for those variants. And so that's where our conversation starts. And this never end because he said, oh, I'm not coming to City Hall, but go to Memphis. And I feel kind of disappointed, but I think your scientific mind, we keep just exchange. And when opportunity comes, he gives this great opportunity to me. And I take this as a huge honor. And he so thoughtful, try to make this true. And he said, actually, we should set up a pre-meeting, talk about, I want to tell you how I can make this event work more successfully. So he set up a half-hour meeting online and told me everything about this program. And I feel very warmed. And he gave actually a pack of tea to me in the airport, said, this is a gift for me. And this is extra, of course, I'm not encouraging everybody to do that. But I feel so warmly welcomed to come to this. And I feel a great honor to do this. And also a couple of words about City of Hope. Some of you may think is a city, actually this City of Hope is a medical center that located in the city of Duarte in Los Angeles. And we have 100 years of the history, and that in history, we made insulin, we made human as the monoclonal antibody as a drug, and that created lots of the royalty to support the institute. So there was like $5 billion endowment to make the institute financially very strong. And it's a very much expanded. When I was there in the first, it's only 2,700 employees. So now it's 130,000, 13,000. I mean 13,000 employees, and it's a national chain of the hospital in Phoenix, in Atlanta, and also in Chicago. So there are 5 different major hospitals, and also the research institute called Beckman Research Institute with 166 researchers there. So I lead one of the cancer center program, which is ranked exceptional. Also, the U.S. News ranked City Hope as the top 5 cancer center. So today I'm going to talk to you, introduce some of the basic ideas, concept to you, about Okazaki Fragment Maturation. And I think this is a source for mutations and life or days. Either live or die, or you have to come mutation, evolve your genome to survive. This is one of the challenges in the clinic that drug resistance. So we made lots of good drugs, including therapeutic drug like immunotherapeutic drugs, regimens, and also targeted drug for the last a couple of decades. The targeted drug has been very powerful works. For example, in non-small cell lung carcinoma, which is a very great disease, but we made a very good drug against tyrosine kinase, especially EGFR tyrosine kinase, is the first generation of the targeted drug because it's the mutation cause the gain of the function. So the tyrosine kinase activity was in very basal level. But when you have mutation, the activity goes up and it's activated all the oncogenic pathways, including many of the genes like KRAS and downstream genes, and that's how cancer started. So if we block EGFR tyrosine kinase activity, so it's a very straightforward strategy that we can make the inhibitors to block kinase activity, and it was very successful. You can observe the cancer shrink a lot just within first 30 days of the clinical treatment with EGFR Taglisol, third generation of the targeted drug, which is very effective. The problem is the drug resistance. Within 2 years, 90% of the patients develop drug resistance, and actually cause metastasis. Now this time, lung cancer migrated into the brain, and within a few weeks, the patient is gone. I had a couple of relatives and also my colleague actually gone with non-small cell lung carcinoma metastasis to the brain. And it's a very bad and common phenomena is observed by clinicians that resistance is associated with they call the mutation burdens. So if you sequence the whole genome, you have lots of mutations, and also that means the cell, a few of the cancer cells survive and they expand, but the genome has been changed. So these three phenomena, drug resistant, the cancer cells survive and the mutation burden is very closely related. So starting from this, I want to introduce you also the DNA replication concepts. So many of you heard, of course, learn this in your college. It's in the textbook about DNA replications. It's about the polymerization of the DNA from the primer, right? And that's true for the leading strands. Polymers always go from 5' to 3'. But the other strand, called the lagging strand, is in opposite orientation. Now the polymers also go from 5' to 3'. So it's like one polymers go from one direction, other polymers go the other direction. Now that causes the problem, and the observation is the DNA replication fork always move from one direction. Now lagging strand need to synthesize in opposite direction. Actually, it make a move. But the thing is that they synthesize in discontinuous fashion, fragment by fragment. Each of fragment has to have a primer, RNA primer to start with de novo synthesis. And then you have another DNA polymerase called DNA polymerase alpha to synthesize a short stretch of DNA with additional errors because polymerase alpha has no proof leaving function, just like HIV reverse transcriptase, no proof. So introduce more mutations. Now the polymerase delta get on high fidelity, has proof leaving functions to go on for synthesize a longer piece of DNA. Now there is no ligase, can ligase RNA DNA together. So this RNA has to be processed. Actually, in fact, some of the alpha synthesized DNA with the errors also processed out. So this process is called Okazaki Fragment Maturation to remove the RNA primers and some of DNA, and then for polymerase to fill in the gap, and for ligase to connect these two pieces of DNA together. So this is a process, and this process could introduce a lot of mutations. So Tom Cantor actually is the one that counted the mutation each of the polymerase can introduce. And he, I think, he divided this polymerase based on their degree of the fidelity into 3 different families. And so the polymerase data and And epsilon is a work family of family B that is a high fidelity, has true linear function, but you still have mutation frequency like one of 10 million. Right, you have mutation frequency. So in normal DNA with good polymerase, you have mutation frequency. But in other families, it's like 100-fold increase. So that Paul alpha have no proof linear function. You have 100-fold of the background mutation rate. So frequently introduce mutation. So you can imagine if polymerase alpha, just like DNA fragment, is not edited out, then you incorporate lots of mutation in general. Of course, we have a repair system, especially mismatch repair is responsible for remove all this even after DNA, newly synthesized DNA is completed. The mismatch repair before histone is assembled. Mismatch repair system can edit out all these errors on the mutation. However, even this polymorphism, we are not thinking about the structural mutation yet, and that will be my next topic. But we are thinking about even the polymorphisms in the yeast, so don't have a division of genetic mutation or somatic mutation. Yeast polymorphisms, when they look at the whole genome, the mutation or polymorphism distribution is not even in the whole genome. This polymorphism is very central in the junction of the two organic fragments. So this is the junction regions that it's very easy to incorporate mutations. Why? Because polymerase alpha, because of the process of the editing process is competing with the transcription factors, with the histones that is going to be assembled this. If it's once assembled too quickly, then all these errors, the shielded, is exclude the nucleus to edit them out. So that's the region you have the peak of the polymorphism frequently happens. So the front nucleus, as I mentioned, is a player very important role. And this protein I spent last 30 years. I was one cloned this gene and published nearly 100 papers on this protein. So we studied structure from crystal structure to mouse model, all the functional mutations identified in the tumor. And I generated 13 lines of the mouse with different cancer mutations on these genes, even though it's rare. But it's a unique feature. This is also represent a very completely different family of the nucleus, because the nucleus in your mind is a distribution enzyme that is used as a tool for DNA clone, right? It recognizes a small motif of DNA sequence and then make a cut. So those are the nucleus. This only exists in certain type of bacteria that we will just use the tools. The most of the nucleus identified in eukaryotic cell is structure specific. They don't recognize the sequence, but they recognize the abnormal DNA structures. So most of the time when we draw DNA structures, it's two lines, but you have different structures. So typical structure is a flat, because in the lagging strand DNA synthesis, the polymerase data actually has a function is not very much recognized by the scientific society. Is called displacement synthesis. So the polymers go on the gap. Once they reach to the last primer, it can displace the either primer and synthesize the gap. So now form a flat structure. Now this structure is needed to be removed. And it's removed by frag endonuclease activity. So this is a typical structure for frag endonuclease to remove the single strand part of the frag. So yeast is a very good model. So that's one nearly 30 years ago. Coronado was the first one knock out the bracket workers in the East. And I actually before that, 1 year before that, there's a science paper by another group that say, oh, Pheromone is a mutator gene. What the mutator gene means, when themselves has mutations, functional deficiency, that cause a whole genome mutation increase like 100-fold. So typical genes are mismatch repair genes. If they are deficient, that cause colon cancer, but the colon cancer is because of accumulation of the mutation. So that's the mutator phenotype. Seriously, but the Pheromone knockout also causes 100-fold mutation. But in Coronado's paper say, oh, but this mutation is not point mutation everybody think about. It's a structural mutation. He said that this 5' flag, if it's not removed and still survive, some still survive is because this flag is incorporated into genome by duplication. Now you have three strands. So this is part is repeated but incorporated into genome. So by sequence, it's a serial paper. I think with one figure, just the sequence motif that he identified, and with one table, and with two figures. The other figure indicated this gene actually synthetically, so it's rather 51 and 52. So because he hinted that the frag is not removed, lots of cell dies because of double strand break. But if you have double strand break repair gene like that 51 and 52, then you can back up a little bit. Okay, so that's essentially so the concept that he already made 30 years ago. He still work on this. And we added on this because fragment endonuclease can also have exonuclease activity. If you have mismatch in the double strand region, the fragment endonuclease can go on to remove all this mutation. Because he said 70% is incorporation of the additional frag, the duplication mutation, but 30% is the point mutation. So we asked them where the 30% of point is come from. Because the was responsible to remove some mismatch. If this mismatch, it can go on. So this is kind of complicated to explain, but just believe me that if there's error, the fellow can go on to remove those errors. So in these junction regions, one is to remove the point mutations as well. This is 30% of total mutation. So this is an introduction summary. This so that's the majority of the reaction components for mammalian gene duplication have already identified, and they reconstituted. But besides the epigenetic control of the cell is not really important. I think so. We published that actual fragment endonuclease. I talk about three major enzymes: polymerase, nuclease, and ligase. All these enzymes know each other when I need to act, when I need to put it on the side. It's by post-translation modification. And sequential modification, like a pheromone methylation, become very sticky to the pCNA. And that phosphorylation is the kicking the fragment endonuclease out of DNA replication. Now the job is done. So sequential modification is very important for pheromone's action. And DNA replication errors are incorporated by DNA polymerase, primase, and poly alpha, which has no proof leading function and have 100-fold more errors. In mismatch repair system, excise errors genome-wise of the delta 3' fragment and 3' nucleus and 5' nucleus mediate the proof leading function. In normal genomes, the polymorphism frequency is peaked at the junction point of the Okazaki fragment and the center of the nucleus, because the nucleus shared the nucleus to excise the mutations. So the center actually is shared, but the center actually is also in the junction point. So these three things: peak of mutations, junction point, but also the center of the nucleus is peaked together. So this is the second point I just mentioned. The third point is formation of the 5' thread DNA is as an intermediate structure for Okazaki fragment maturation is a major source of genome instability. It may result in double-strand break and cell death, or be incorporated into genome as duplication mutation for cell survival. So this is the three take-home message to set my background of this talk. Now I want to say what happens if a fragment endonucleus is knocked out. And so actually this is a third phenomenon. Like I mentioned that there are two phenotypes already. One is the mutated phenotype. Fragment endonucleus knockout has a 100-fold mutation increase whole genome. Second is that it is synthetically soluble with the lead 51 and 52. Third phenotype is a conditional lethality. Knockout of the yeast cell still can survive in 30 degrees, which is normal temperature we use to culture the yeast cells. But if you put the plates into 37 degrees, we usually grow E. coli and also mammalian cell, the cell dies. So also in his paper in 1997, he said, oh, this is conditional lethality. So this is three phenotype he characterized. But this happens to quite many other genes as well. What so let's stop there. But in 2019, one of the first one from the that she left the place, in the mutant place in 37 degrees for too long. She forgot about even the agar become very dry. But she observed it's very clear that some of the cells she spotted there can grow in 37 degrees. It's compact. So it's a revert. It's a revert. Some cells supposed to die. It's compact. It's a revert. Now we can take advantage of the sequence. We can sequence the whole genome to find out we had under assumption that some of the genes may be overexpressed, but we were looking at genomic DNA of the sequence. We found actually polymerase mutation, polymerase data I talked about is doing the displacement synthesis. Actually, all data had a mutation. So that is the first observation on the sequence. I try to explain the DNA replication stress because everything, 37 degrees is a DNA replication stress. I think we don't know exactly how high temperature gives cell stress, but there are many factors, and include genetic factors. Some of genetic deficiency is a DNA replication stress. So DNA replication stress is the concept of DNA replication machinery is very tight. So if you have one component has mutations, of course, and especially in cancer cell, the replication demand is so high. Lots of times it's a polymorphism, right? So you have multiple sets of DNA to replicate, and the cell divides so fast. So this is the replication stress. I indicate a sick person is a packing, and the pathway is also not very good. So DNA can have a lot of secondary structure, serve as a barrier for DNA replication, difficult to replicate the lesions. That's DNA replication stress. So that's I wanted to make sure that you got this very abstract concept. How cells with DNA effects survive replication stress. Say I want to put this cell culture in 37 degrees instead of 30 degrees. So we found, I think I don't have a pointer. So this, with this gene knockout, the problem is this knockout. After while this cells can grow. And apparently this is from a single cell that the real cell survive. So we pick up this colony and grow in the medium. And now if you immediately put it in 30 cell nuclei, they can grow just normal, just like white type. So it's not to go back to the active time. And they kept the genetics, that's genetic reverse. And when we sequence lots of mutation is on the polymerase. Remember, we are sequence only the survived cells. We don't know what happens in the dead cells. But I also told you that mutation occurs in the whole genome-wise. It's not like a God, you just change this figure. And no, it's not only changing polymerase. Lots of cells has mutations in other regions may already have. But the majority of this polymerase mutation has called internal tandem duplication mutation. About half of those very common is internal tandem duplication mutation. And so when we put those mutations, so now we can do like site-directed mutagenesis and incorporate this back to replace the wild-type polymerase. Now this mutant, double mutant, put back into lat 27, which is the fragment mucus gene. In those mutant cells, you put the polymerase gene back. Now all the cells can survive. Just like the white. So this is the backup. Now the failure is still deleted, but if you can put the polymerase mutation, that they can go in certain degree, just like white. Okay. So this is the phenotype, and so by chemical acids, we indicate. So this is I not introduce this chemical acid to you. But what we follow is that this polymerase lost additional function. Now I said that one function loss give your genome-wise this kind of structure. Now the polymerase lost the function to displacement sensitive. So this is no more fragment. That is pattern solving problem. The problem on the fragment are these double strand. Now no more fragment, no more displacement. So this is a synthetic survival, right? Two mutations added together, cell can survive. So this is the what we see in biochemistry. And the mutation spectrum looks at no more duplication mutation, but there's a point mutation there because this structure is gone. So you have these, but the polymer is not removed. The anode incorporated by polymerase alpha is not removed. So this incorporates into genome. But I told you that it's no magnets can give iron. So my prediction is now iron H, both iron H become essential. And then we tested this and we get approval for this hypothesis. I said, oh no, yet. So this is a short story about this phenotype. There's a next question we want to ask. Because now when we look at this revert, we also see some of the duplication issues. How can I tell you? I think I missed something here. But this duplication mutation is different from the lat 27 mutation anymore. Because the lat 27 mutation is a duplication, means repeat of the two fragments of DNA exactly same duplication. Now what we see in the mutation spectrum is a two duplication mutation, but there is a spacer sequence in the middle, which sequence people usually don't recognize. They characterize this kind of mutation as a duplication mutation. Middle space leading. We can explain why this comes from. So this is a characterization of the alternative mutation compared to the classical duplication mutation, we have a spacer region. Where this spacer region, this spacer does not align to any of the sequence. If you align in the same direction, you align this sequence in the same direction of genome, does not these things. So we did several patterns of this. This is one of the patterns that we can see that this is the spacer sequence. This is the duplication sequence. This spacer sequence actually is incorporated by 3' frag. That 3' frag is different from 5' frag because polymerase now can extend the 3' frag. Especially when you have some of the complementary sequences, they can form the hairpin structure, and the polymerase can extend this gap. So that's why you don't align these in the same direction anyway. But because it's a complement to your own strand, so this sequence, so I think there are many of the sequences in the genome, in different genomes, and people just not paying attention on this. Our science paper is not very scientific at all. We think it's a very good piece of work, but they don't understand. I mean, even in the end, after several revisions, but still. So it's not a popular concept, but this is true, I think. Did you get it now so far? Good. So this kind of result predict that you have 3' frag. You have converted 5' frag to 3' frag, but the 80% you have to demonstrate this. So now. Well, we are talking about this part is that try to solve and bring a kind of double strand 3' end, but the only frag end can extend, can be labeled by the activity is terminal. So double strand 3' end is blocked by one of the enzyme, but the fully single strand end is left alone. So it can be labeled. So that's and this kind of reaction, they only show the applications that can be 7 mutation fragment that's not out, but also in 37 degree, not in other genetic background or any other conditions. I'm not sure that it's very specific. So this is a very good essay. Actually, we are further developing this PCR based method. So what I also told you so far is that how alternative Okazaki fragment maturation survive the cell. So there are quite a few ways. One way is that so I was telling you, you must be confused. The polymerase incorporated mutation lost the function. So that survives, one way to survive still, right? But how polymerase duplication mutation incorporated, and this duplication mutation is different from classical duplication mutation because of 3' frag. And 3' frag make you imagine many of the possibilities. One way is you can just remove the 3' frag. The other way is from secondary structure, so you can see the mutation spectrum, mutation spot left over in survived cells that have duplication mutation. Actually, there are other possibilities that we are investigating. Because 3', this is the intermediate structure for recombination that you can easily invade to the sister chromatin and it can extend and then come back. So through repair this by recombination, right? The other possibility, actually, we observe very exciting is 3' actually can go to align with the opposite strand of the sister chromatin. And this, you can see a lot of different mutation spectrums. But all these ways is a way to make cells survive. So the last question I wanted to tell you is could this reversion process be inventionally managed? And that's it's relevant to what I raised in the first place, drug resistance. So if can we prevent drug resistance, or can we reverse drug resistance by managing those kind of DNA replication errors? So we found actually in this process a checkpoint gene activation is very important. Essentially, the cell so it's 3' demonstrated. We only culture cell for 4 hours in 37 degrees. So cell is struggling for its survival to make this happen. The conversion from 5' to 3' happens. But in this time, you need a cell to be arrested a little bit, so you have time to do this. So the checkpoint is very important. So it's the gene called DAN1. Actually, it's the only homolog of the CHEK1 and the CHEK2 in human genes. So DAN1, now if we inactivate this gene, now you don't have time to do 5' 3' conversion, to do the mutation, and to do anything. So if we knock this out, we don't see the 3' flap anymore. We don't see the alternative duplication mutation anymore, and we don't see cell survival anymore. So this is what we talk about. So activate the cell cycle checkpoint gene and one knockout lead these 3 different consequences. So this is a summary about this paper. I will leave sufficient time for you to ask a question if you have any. But so far I told you is gene risk and cellular disability are originated at the DNA replication fork due to various mutation, stress force, block of double-strand break, repair pathway generated at the S-phase, and at the S-phase results in the synthetic lethality, which is exemplified by the current mainstream cancer therapy, PARP inhibitors. PARP inhibitor, it's only work. So in this model that we have quite a bit of data, and other people have published. PARP is the PARP1. It is the enzyme initially identified as a base excision repair. So they think PARP inhibitor is making the PARP to cut onto the nick place in the G1 phase for base excision repair. G1 phase you have a nick, then PARP will binding onto the nick site and make a modification on histone for base excision repair pathway to set in and to repair this nick. But now you probably realize there are many nicks in the S phase because of the Okazaki Fragment Maturation. You have this short piece in whole chain, many more. So nick is only occasionally occurs in G1 phase. It's caused by DNA damage. But in S phase, you are spontaneously generate all these nicks. So PARP1 is main function in S phase in binding to those nick, binding to those flap site. So now, if you block PARP, two things, you block the activity. Now you don't have the function, so you cause the replication fork problem in the first point. Now, BRCA1, BRCA2 is involving double-strand break response repair. I call it DSBRR. Now you have this point. Another point downstream. I mean, in this risk pathway also blocked. Now cell is going to die. But I also told you now today that there's an alternative pathway for Okazaki Fragment Maturation, which is 3' flap. So we can actually develop inhibitors to block that way too. So it's a triangle, right? The replication fork is one point. The other point is double-strand break response to repair pathway. The other fork is alternative fragment maturation. So these actually offer us more opportunity for cancer therapy. And we are putting quite a lot of effort in doing this. And so one of our current focus is to test the synthetic restorative relationship among Okazaki Fragment Maturation enzyme inhibitors. PRCNS means genetic defects like PRCA1, PRCA2, but it's involved in the testing. So I screened a CRISPR library for all 365 genes involved in double-strand break and other repair pathways to test the PARP inhibitors. So at once, it's a library that you can test at once. So we screened out 3 very striking genes out of the 365 because based on current knowledge, say the genes involved in the double-strand break response repair could also serve as PRCNS genes. So NCI called a meeting like a couple years ago, trying to broaden the therapeutic window for PARP inhibitors. So if we can identify additional genes, but every gene is like more PRCA1, PRCA2. So which genes really count as a PRCNS is a question. So that's where I started the screen like this. And some genes actually make a cell more resistant to PARP instead of to be more sensitized. So I think it's will result in some very meaningful data for clinician, but whole, when they identify, so every single of the patient was sequenced for their therapeutic windows. So when they got the sequence result, they come to ask me 2 questions. If this mutation count, because this last time they identified a point mutation. So in which domain, which functional domains can disrupt the function? Because PRCNS is only disrupt the function, it's count for PNC. The other question is actually which PARP inhibitor can we use the best for this patient if this mutation count? So there are like 200 PARP inhibitor has been in laboratory, preclinical study, and clinical use currently. So there are quite a lot of choices, and the mechanism probably each inhibitor is slightly different. Currently, there are 2 different mechanisms available. One is block the catalytic activity of PARP. The other is make the PARP chop onto the DNA nick or flap side. Now it's caused even a more severe problems because the protein a lot of times is covalently linked to DNA. So now you need to repair this kind of. So PARP inhibitor is very different. So these are 2 questions that our precision medicine program, they come to the consultancy. So this is some of the conclusions that I want to take home, and I feel also very appreciated that some of the appreciative patient that they make donations to us. And 2019, non-small cell lung carcinoma patient they come to us through this doctor, and he has EGFR mutations, and the tyrosine kinase inhibitor Tagrisso worked very well on him, but he said, I only have 2 years life. I make like $38 million revenue for his textile industry, but I want to spend some time to extend my life. So do you have some kind of research going on? I have been working on this for many years, but he said, you have to go down, get it down within 2 years. Otherwise, I'm gone. So we started this. We tried to screen, use the synthetic restorative theory, try to prevent drug resistant or reverse. So Mineroid is a one that now currently in the clinical trial, and he enrolled in this. So since 19, so it's 6 years past. He still survives. So I feel very happy. So there are like 18 patients so enrolled in this program. And finally, I want to thank the group I told you, some of you, I had many postdocs went through my lab, 6 year postdocs went through. And I'm still looking for postdocs to join our laboratory. This effort, even I'm well supported. I have no complaint on the but it is also I'm well supported by City Hope. We want to get translational research done. So, but I always tell my postdoc, I am like a bank. Myself has no knowledge because your comes, you brought different expertise, different knowledge, and I bank and as I transfer to next postdoc and make them, to generate some of the very useful knowledge. So these. And I also want to thank my collaborators that they provide lots of good ideas. I also feel so very much appreciative of Dr. Williams this morning. He did so much homework. He established this database, but also did homework on my genes and generated some very useful data. I feel very excited. I feel like this best place. I have given 112 seminars in different campuses, and this is the best experience I ever had. Everybody is like you talk to a real person, talk like your close colleague, talk like to your old friend. Yes, I never got this kind of feeling before. But thank you very much for your attention.
I think it's a very strange question. In just thinking about the process of replication and the Okazaki fragment, it seems like it was a sort of risky endeavor. So what were the limitations in terms of design that prevented cells from coming up with a better, less error-prone method?
So, it's a risk if cell death is not a problem, right? It's not cause cancer. So what we worry about the cell survivorship. So it's not I want to offer strategy how can make cell survive. I want to make cell death is fine. But if you want to live, you live a normal way. So that's it is kind of thing. So I will not be exactly prepared to answer your question. The strategy is, if one either kills it, but I think the problem is the mutagenesis, all the gene replication and process can incorporate mutations. And polymerase can input, but the repair system can remove. Some of the repair system is error-prone, which also causes the problem. So mutation is that makes cells survive, but in abnormal way, very aggressive way, very low fidelity way that transform the cell into cancer.
Yes, I have two questions. One is sort of basic molecular biology that are probably missed. So in forming the nucleosome, you talked about there were 3 conditions that you had there for the nucleosome, sort of at the core of the nucleosome, the Okazaki fragment junction, and I forget the third one.
It's the peak of the mutation.
So does that result in a consistent signature across all cell types, or are the nucleosomes very different from one cell to the other, or one cell type to the other? Or can you say, oh, I'm reading through the DNA, and I can tell that that's probably where nucleosome will form. And therefore, I want to look more closely at that region for mutations. Can you do that kind of at a global scale?
So it's a 2015 series of 6 papers from 3 different groups was published in East Genome, Okazaki map, nucleosome center, Okazaki map, the frequency of the mutations. And the mutation here, I'm talking the point mutation, they call it point marker mutation, not the mutation. And they found that, and also some of transcription factors can block these also. So transcription factor binding is also another factor to think about it. And that the junction, they were able to map the junction where is the genome. So that's the first time we were able to do this map. I'm not participating as a group on this, but nonetheless asking if this can also come in mammalian cell. And we have not done this. We were using yeast as a reference. Don't take it wrong, because I'm not a yeast person. I just use this model. But our aim is to translate this data into the mammalian cells, so we can see this. But now I'm more interested in the structural variants. So, especially how, like a genetic mutation, say FAN1, say BRCA1, say mismatch repair, how they cause the somatic mutation which contribute to cancer. There's a correspondence in this, because the yeast data is only limited to the point mutation, and there's no surprise at the junction, as I explained, because there is no opportunity, more opportunity to be incorporated. There's no more opportunity to be missed out to remove this. That's why the peak is arrived on the center point of the mutation.
Did anybody else have a question before I ask my second one? Because I don't want to be a pig about asking questions.
No.
Enza?
No, you can ask.
Okay, I'll be a pig then. So your entire presentation implicitly focused, well, it explicitly focused on cancer and implicitly focused on mitotic error. And I'm just, so Enza and I and the team are looking at meiotic mutation rates that are germline. And so I'm just wondering, obviously a lot of cells in our brains are basically post-mitotic. A lot of cells are post-mitotic and hard muscle. The cells that make sense for you to focus on are crypt cells, etc., where we know that EGFR is super important in terms of cancer risk. I'm just wondering, the mechanisms you talked about, Binghui, to what extent would they have any impact on post-mitotic cells? You know, everything, because all the money is in Alzheimer's disease now, everybody's going, I wonder if Okazaki fragments do anything to Alzheimer's disease. But so somatic versus germline.
Right. So I hope everybody understands what Dr. Williams is saying. Indeed, I was very much focused on genetic mutation in my project. The double copy of the chromosomes. Not if it's in the germline cells, sperm development, is one copy of the DNA. I think mutation mechanism probably is very similar, right? And but I think he's more leading to what is the influential of the germline mutations on the cancer, like especially. And I'm very interested in this. I say the FAN1 mutation in germline, that causes somatic mutation on duplication mutation, vast majority, right? 70% will be duplication mutation, and which we observe is frequent in leukemia and other many cancers, prostate cancer, that the duplication mutation is there. But is this attributed to the germline mutation of the FAIR started from day one, that because this mutation, so some of the individual has more frequent of this mutation, which lead their early onset of the cancer. So I was asked around the so-called expertise, but I think I found a way today that how the germline mutation influence the somatic mutation, which result in cancer. I think this is my next project I want to start, but I feel like my knowledge is very limited, and I have not found a conclusion. I want to further explore this Johnson.
I sent you a DNA2 analysis, but I wasn't doing email here. I was actually mapping it.
Yeah, so DNA2 is another gene we have been focused on in the last 30 years. People just say, oh, being young, so focused because in all your careers, focus on two genes, FAIR1 and DNA2. So in all different kind of organisms, when one postdoc comes different expertise, I say, oh, why don't we look at this aspect? And actually has been very good. So I mean, these two glands, it's a sixth cycle on it. It's a 31st year. Question from students, you treated me so well, so you got to give me some praise.
And actually said all kinds of nice things about them and told them to interact and talk,
Yeah.
interact,
Yeah.
and ask stupid questions. You want me to ask a stupid question just to give you a demonstration?
Like, if there's any topics that didn't make sense, you want clarification.
I was wondering, like, since the mutations are from this part to this part, can it be reversed back to the original state and just like just take out that mutation?
Very good question, but if 5' flag, still survived because the converted to sleep, can this reverse back? I think that's the question. But the problem is that because we don't have 5' flag and it's removed, so we found an alternative way that now 3' flag and occurs can be removed to solve this. So it's not like so NIH will not take this as a priority for you to see if there is a this fold back kind of question. Because I made a program, this cannot be used. So I said, I told NIH, oh, there's another way that we should, and this experiment drug resistant because it's alternative pathway for cell to survive. So what is the mechanism? How this was processed? If this invade other strain, so this kind of question I asked.
Okay, thank you.
When a salesperson served for a very major biotech company, walked into my office. He said, "I'm not selling anything to you, but you remember you gave me a class, many years ago, and then this still benefit for my job." I feel a little bit mixed feeling that I actually you never know, some people can be very long
So I guess if I understand, it's kind of like a war game. So you try and figure out if you can inhibit the process of OFM, then perhaps you can get the cancer cells to fail to repair the damage, go into double-stranded break, and then die. But is there a risk that in that process you also are introducing more mutations, and so then you're creating, so then you have that the opportunity for the cells to evolve and become this?
It's very similar feeling I share with Megan that if you do something, then you have another way to back up. So it's like a never end, right? So I think epigenetic factors will be better targeted. It's not a, we were, for many years, we were looking for specific type drugs. One gene, one drug, one pathway, and broad cancer progression. And that's what happened to EGFR tyrosine kinase inhibitor. Indeed, worked, but never worked. Now, I think epigenetic factor, they control the, I think more process. And this is the project ongoing in my lab. I feel very, very excited. It's a change in my concept. I must spend a lot of money from institute to try to develop it. We only, had one clinical trial, which is it's like not so novel. It's a combination therapy, and I don't believe it will solve drug resistance problem at all. I mean, at once, but epigenetic factor, I think is a way to think about it. So I because of the firewall work, I try to identify arginine dimethylase, and which is not there like 10 years ago when I started. So, everything has a transferase and then J enzyme, it's the enzyme to remove it. So, but the arginine dimethylase are not there. So it's a new enzyme. But I think this introduced a lot of good science into it. But I think it's also very promising to develop new ingredients. So I'm recruiting more people onto this. It's develop an inhibitor to clinic. It really takes a long way. I have ever been excited about this piece. It's like, now I want to work with since nobody is there. I also feel like my time is limited. Right? I always keep telling people, oh, I have 10 more years. For the last 10 years, I say I have 10 more years. But I indeed, my career, but I'm very healthy and very active, but life space. So I wanted to use my wife said, oh, I want to see grandparents. I said, it's good. So I can come home like 8 o'clock, 9 o'clock. Yeah. Thanks again for all your questions.